Hello everyone, welcome back to the channel. I'm Zach Hancock, an evolutionary biologist that specializes in population genetics. This channel has been around for a little over a year at this point, and recently I've been kind of reflecting on some topics that I tackled early on. My second video ever was this one, the onion test and genomic functionality. It's got a whopping 400 views and is just a PowerPoint presentation of me talking and sort of laying out my case for junk DNA. The audio is pretty terrible, and the entire thing was recorded in one take with literally no editing. Now those of you that follow the creation-evolution debate scene and are perhaps frequent readers of the often unhinged Discovery Institute's blog Evolution News will likely be aware that they have been devoting a considerable amount of time and brain space to a friend of the channel, Dr. Dan Stern-Cardinale from Creation Myths. Dan recently had a debate with Casey Luskin over at the Non Sequitur, link in the description below, over the existence of junk DNA in the human genome. This debate has led to several responses and counter-responses, both from Dan on his YouTube channel and several from ID proponents, both in Evolution News as well as on their Discovery Institute podcast. What precipitated all of this was a long story short video produced by the Discovery Institute on the history and existence of junk DNA that Dan responded to. We're going to touch on some of the claims of that video throughout. We're not going to review it or point out specific things, but we will be addressing many of the things, if not all of the things, claimed in that video. But to suffice it to say, the video included a stunningly inaccurate view of both the history of the junk DNA hypothesis as well as the evidence supporting it. So with all this drama going on, and given that we're, it's been over a year now since I put out a video specifically going through the evidence for junk DNA, I thought that this would be a good opportunity to sort of revamp that original video. So in this video, I'm going to provide for you the real history of the junk DNA hypothesis and the original evidence supporting its existence, both from a theoretical and mathematical perspective, thinking about why junk DNA is a necessity irrespective of if you believe in universal common descent or not, and try to deal with the sort of philosophical and methodological confusions that have emerged between not just creationists, but molecular biologists and evolutionary biologists about how we think about function to sort of help us understand why it's so easy to find papers in the secular literature proclaiming the death of junk DNA. So this one's going to be exhaustive. We shall leave no stone unturned, so grab yourself a beer, some popcorn, whatever you need to relax and get comfortable, because this is going to be the complete history of the junk DNA hypothesis. Pictured here are two species of shovel bugs, small, beach-dwelling crustaceans that are closely related to roly-polies. These two species are within the same genus and are no more than 5 to 10 million years diverged, about as diverged as we are from chimpanzees. On the surface, they look strikingly similar. Indeed, they have the same sort of ecology. They're both blind, deposit-feeding organisms that live in marine environments. And yet there's something remarkably different between them. They differ in their total genome size, that is, the amount of DNA they possess, by over 10 billion nucleotides. Can you guess which of these two has the bigger genome? No? It's this one, the Canadian shovel bug, which clocks in at a whopping 14 billion base pair genome. That means if you laid their genome out like a ladder and counted the number of nucleotides, or the rungs in the ladder, it would be 14 billion rungs long. For comparison, humans only have 3 billion rungs in our DNA ladder, which is close to the number of rungs this species of shovelbug has, which is endemic to Louisiana and North Texas. How do we make sense of this? The genome of an organism is often described as a blueprint, is a layout for how to build that organism. It seems like the same number of lines of instruction are needed to build a human as is needed to build this shovelbug, but an additional 10 billion lines are needed to build this shovelbug. Genome sizes vary immensely across the tree of life. The smallest genomes are in viruses, followed by most prokaryotes like bacteria and archaea. Unicellular eukaryotes, called protozoans or protists, typically have much larger genomes, but usually smaller than many multicellular eukaryotes like shovelbugs, onions, and ourselves. But there are many exceptions to this rule. Amoeba, for example, are famous for having monstrous genomes, exceeding tens of billions of base pairs in several instances, despite being just a single-cell blob. They have no development, no tissue-specific gene expression, no organs to build, no complex behavioral networks, no sociality, and yet they possess many times more DNA than a human, an arguably more complex being. Indeed, the trend of genome size with complexity decays rapidly once you start comparing multicellular animals and plants. An onion, for example, has a genome size 5 times larger than ours, while a pufferfish genome is much smaller, only about 500 million bases. A fruit fly apparently only needs 180 million bases to be built, but a mosquito, a related dipteran, needs 1.5 billion bases. A recent study completely sequenced the genome of the lowly krill, the crustacean famous for being eaten by baleen whales. Its genome is 48 billion bases long, 16 times bigger than ours. A natural question emerges from these seemingly paradoxical observations. What is all this extra DNA? In fact, for many decades, this observation was known as the C-value paradox, which described the fact that there appeared to be no clear relationship between organismal complexity and the amount of DNA they possessed. Of course, this is only partly true. As I already said, viruses and bacteria, much simpler organisms than eukaryotes, have much smaller genomes. So there is some relationship with complexity, at least at the highest levels of organization, but it disappears entirely once you start comparing multicellular eukaryotes like insects, plants, worms, birds, etc. If we insist on the analogy of the genome as a blueprint for building an organism, then we need to answer the rather bewildering question of why there should be so much DNA in the Canadian shovelbug, but so little in its close relative, the Texan shovelbug. The biologist T. Ryan Gregory framed this oddity in what he called the onion test, particularly in how it can relate to the junk DNA hypothesis. It goes like this. The onion test is a simple reality check for anyone who thinks they have come up with a universal function for junk DNA. Whatever your proposed function, ask yourself this question. Can I explain why an onion needs about 5 times more non-coding DNA for this function than a human? And importantly, the test is not just a comparison between onions and humans, but between onions and other onions. He goes on, further, if you think perhaps onions are somehow special, consider that members of the genus Allium range in genome size from 7 picograms to 31.5 picograms. Picograms actually equals gigabases, or billions of bases, so from 7 billion to 31 billion. So why can one species make do with 1/5 as much regulation, structural maintenance, protection against mutagens, or insert your preferred universal function as another? This is the same sort of question as it pertains to shovelbugs. Why does the Canadian shovelbug need nearly 5 times more DNA for regulation, or whatever non-coding function you think it has, than the Texan one? As we discuss the concept of junk DNA, going through the history, the original arguments for it, and the modern biochemical evidence for it, I want this observation to always be in the back of your mind. Anytime you think, but maybe this level of transcription or histone binding is actually necessary, but we just don't know why, Ask yourself, why should two species of onion need such dramatically different amounts of DNA to do the exact same thing? We're going to return to the C-value paradox, and indeed we're going to discuss how it was resolved. But first, we should begin by clearly defining what we mean when we say junk DNA, and in particular, the different conceptions of function in biological systems. In biology, there are two key conceptions of function that have sometimes been used interchangeably in the literature. These confusions of function have had a ripple effect across every discussion of junk DNA and human genome functionality, and so it's critical we are very clear on what we're talking about. The first concept of function is the simplest, and it's known as the causal definition. This has the form A is the cause of B. For example, a genomic region A binds a transcription factor B. Under the causal definition of function, the function of region A is to bind factor B. Notice this says nothing about why region A binds the transcription factor, or how this relationship between A and B came about, or how it's maintained. It is a simple causal statement. Many molecular and cellular biologists favor this definition of function because much of their work is descriptive. They want to understand how some specific biomolecule behaves in the cell, and so they will describe it mechanistically. A interacts with B that causes C to fold and ultimately bind D. This sort of descriptive work is very important for understanding what is going on in the cell, but it doesn't tell us why these things are happening, or even whether these things are necessary to occur. Indeed, the causal definition of function can lead to silly things. A common counter to this definition, first used by Dan Grauer, is to ask the function of a shoe. If you're walking down the street and you step on gum, your shoe may bind to that gum. Since this is a thing that your shoe does, you can describe it in a causal way. My shoe binds gum, hence the function of my shoe is to bind gum. Notice it has the same form as genomic region A binds transcription factor B, so the function of A is to bind B. But shoes were not designed or maintained to bind gum. This is a side effect of being placed on your feet and the carelessness of people spitting their gum onto the sidewalk. So if your shoe becomes so degraded that it no longer protects your feet from the hot sidewalk or from a rocky path, it may still be able to bind gum, but you're more than likely to say that those shoes have lost all function. This brings us to the second definition of function, and it's the one that's the most relevant when we're talking about junk DNA. This is the selected effect definition. It asks, why does genomic region A bind transcription factor B? For genomic region A to be functional under the selected effect definition, it must exist because selection first favored its existence and is subsequently maintaining it in the face of mutation. Again, shoes were first designed to protect your feet, and if they degrade such that they no longer do that, they cease being functional under the selected effect definition. They have lost the function they were designed to do. Notice that this says nothing about whether or not genomic region A does anything at all. Of course it does something. It's a biomolecule in an aqueous solution surrounded by other chemically active molecules with ample cellular energy around to catalyze reactions. Stuff is gonna happen. The question is whether those happenings are necessary, whether they were selected to occur, and whether they are protected against subsequent mutations. This last point is critical. If a genomic region is free to mutate in any way and yet still be claimed to be functional, this is akin to saying that there are no negative mutations that can ever happen here, including completely deleting the region. Dan Grauer, in a biting critique of the ENCODE project in 2012, which we're going to talk about in more detail later, compared this to leaving a television set on for 1,000,000 years and expecting it to still be working when you come back. Without constant maintenance, things degrade, and the genome is no exception. Mutations are constantly breaking things, and only selection can purge those mutations. Thus, a major expectation of the selected effect definition is that functional regions should show a sign of constraint. That is, they should have less variation than non-functional regions. We will talk about this in more detail later as well. For our purposes at the moment, I hope that it is clear that the definition of function we really care about is the selected effect definition. That for a region to be functional, it needs to be capable of being broken, of having some fitness cost if it is not present or becomes degraded. Junk DNA is thus all DNA that does not have a selected effect function. It can degrade, be deleted, shuffled around, etc., and it has no phenotypic effect on the organism. Before we get into the nitty-gritty of the history and evidence for the existence of junk DNA, I'd like to take us down a slightly different route. Let's take some time to chat about the ecology of parasites. Often the discussion of junk DNA dives straight into sort of the depths of biochemistry, but to kind of gather an intuition for junk DNA, I think it'll be helpful to start with something we're all a little bit more familiar with. After all, every single one of us harbors parasites of all different kinds, and it turns out junk DNA is really just another kind of parasite. Ecology is the study of biotic and abiotic interactions. That is, how do living things interact with each other and their environment, and how do these interactions shape their evolution? We often classify interactions between organisms based on their impacts. For example, if two organisms are vying for the same resource, we characterize that interaction as competitive, and this typically affects each of them negatively. There also exists commensalisms, in which an organism benefits while the other is completely indifferent to the interaction. There are mutualisms, in which both benefit from the interaction, and of course, parasitism, in which one benefits at the expense of the other. This last form of ecological interaction will be the focus as we try to conceptualize junk DNA in light of ecology. There are many kinds of parasites. There are exoparasites, which sort of extract resources from the surface of an organism. These are things like mosquitoes and ticks, which take their blood meal externally. Then there are endoparasites that actually live within the host, such as tapeworms or blood flukes. These organisms occupy an organ. They may live in your intestines or in your liver, in your heart, etc. At an even lower level than that, there are parasites that infect your cells. Plasmodium, the causal agent of malaria, is a protozoan that infects blood cells, literally living inside of them. Of course, there are parasites that infect yet even lower levels. Viruses are genomic parasites. They invade your cell and then they infect your genome with their own genetic material, hijacking the replication and transcription machinery of your cell to force it to make copies of itself. Typically, your white blood cells will destroy all such cells infected by this virus, but sometimes these viral genomic insertions can persist across your lifetime and across generations if they manage to infect your germ cells. When this occurs, these parasites become what we call endogenized. They have become a natural part of your genome and will be passed on to all of your descendants. In fact, your genome is replete with ancient parasites, most of which are little more than genomic fossils. More than 50% of your genome is composed of transposons, genetic elements that possess, when intact, the ability to recruit cellular transcription and translation machinery to get themselves first copied and then inserted into other regions of your genome. There are many classes of transposons, but they all share a key unifying feature. They exist to proliferate. And this proliferation is akin to spreading an infection. For a virus, infecting new cells is how it spreads, but for transposons, this spreading is done by infecting new genomic positions. If you imagine the genome as linear from position 1 to position 3 billion, the more positions that are occupied by a transposon of a given family, the more infectious or transmissible that transposon is. Now, the impacts of parasites on their host varies widely. Some almost always kill their host, whereas others are so mild that the host may never even have symptoms of being infected. And the extent of the harm may not be constant. For example, a healthy person can get the flu and typically recover just fine, whereas someone that is malnourished may die because their body doesn't have the resources to deal with it. Similarly, the genome has certain tools for silencing and defending against transposon proliferation, some of which are quite elaborate and require special RNA molecules that identify and degrade these rogue transcripts before they can get picked up by reverse transcriptase and reinserted into some new genomic position. No organism is free from parasites of one kind or another, and our ability to tolerate a given parasitic load is a function of how costly these parasites are to us relative to our ability to actually gather enough resources to deal with them, and the relative difference in survival and reproduction between infected and non-infected individuals. When the cost of harboring a parasite is very low, its ability to spread through the population is a simple function of its ability to make copies of itself, and its host's ability to just have children. We're going to introduce the concept of the population genetic environment in the next section, which will situate these ideas within the realms of population genetics, but for now we can think simply of it as a cost analysis. If I am infected by a parasite, will I still be able to find a mate and have as many descendants on average as someone not infected? If that parasite is the common cold, face mites, or the thousands of endogenized retroviruses that call our genomes home, then yeah, nothing is stopping you from having offspring. Those parasites, while they're not free to harbor, cost you so little that they just keep on keeping on irregardless of their presence. To bring this analogy home, junk DNA is a kind of genomic parasite. It covers all the elements in your genome that are there not because they're doing anything for you, the host, just like the mites that live in your eyelashes aren't doing anything for you. This is not to say they're not doing anything at all. Parasites aren't just passive passengers that are just sitting there. They're typically actively stealing resources from you, even if so little you just don't notice. Junk DNA will get transcribed, will bind proteins, may cause folding patterns, and will even sometimes get translated. As an example, reverse transcriptase is an enzyme that must be transcribed and translated, but its sole purpose is to snag random transcripts from the nucleus and reinsert them into the genome randomly. They are coded for by viral and transposon sequences and have no functional benefit to you. In fact, they are harmful to you because a rogue transcript plopped into a protein-coding gene might break that gene's function. Many cancerous cells, for example, show evidence of excess transposon proliferation. If we can begin by thinking about the genome as a kind of environment that can be invaded just like your cells, your tissues, your organs, then we can begin to understand not only why junk DNA exists, but why it's virtually impossible for it not to exist in at least some capacity. It would be like saying that parasites can invade every other part of your body, but your genome is somehow immune from parasitism. But how can we predict how much junk DNA should be in any given organism? That is, can we explain the general patterns of why genomic parasites do so well in some genomes and not in other genomes? And can these differences in proliferation ability explain the C-value paradox we mentioned previously? To answer these questions, let's talk about what famed evolutionary biologist Michael Lynch calls the population genetic environment. Evolutionary biology is ultimately the study of the interaction between a few fundamental forces: natural selection, genetic drift, mutation, and gene flow. Which of these four forces dominate the population dynamics will dictate how evolution is expected to proceed. Since the 1920s, population geneticists have worked out detailed mathematical formulas that exactly describe the expected dynamics of a population under different regimes of selection, drift, and mutation. A key result from this work is that the rate of genetic drift, which is the random change in allele frequencies, is proportional to the size of the population. To understand this, think about a series of coin flips. In expectation, every flip has a 50-50 chance of landing heads or tails. If you flip a coin 10 times, you wouldn't be too surprised to find 80% heads and 20% tails. You differed from expectation by chance alone. But if you flipped it 10,000 times, you would be pretty surprised to see chance play such a large role. You'd expect the distribution to be much closer to 50-50. Thus, in small populations, chance, that is genetic drift, is expected to play a larger role than in large populations. Unlike drift, selection is most efficient in large populations. This is because the dynamics of large populations are more deterministic because they aren't dominated by chance. So even very slightly advantageous alleles can be favored in large populations. Similarly, very slightly deleterious alleles can be purged. But when the population is small, chance can interfere with selection and can cause positive alleles to get lost and deleterious ones to get fixed. Simplistically, selection is expected to dominate anytime the following inequality is satisfied. 1 is less than the absolute value of 2 times n times s, where n is the population size and s is the fitness effect of the mutation in question. Notice that selection can dominate either because the fitness effect of the mutation is very large and the population size is relatively small, or if the population size is very large, but the effect of the mutation is very small. This leads to a very important insight. When populations are very large, selection can see mutations with incredibly small effects. But as the population size gets smaller, the effects of mutations need to get proportionately larger to still be seen by selection. Mutation is also an important factor here. Mutations can either increase the amount of DNA in a cell by duplication, or it can decrease it by deletion. Each of these may have some impact on the fitness of an organism. A duplication is almost always deleterious because it causes excess redundant DNA that the cell must expend energy to copy. This is true despite the fact that new genes must emerge this way. A parent copy must first get duplicated, making this sort of redundant copy, and then subsequent mutations tinker with that copy to generate a new function. But along the way, that duplicate has no function and may even be slightly deleterious to maintain. Deletions depend on the genomic context for their fitness effects. Deleting a redundant gene, for example, might actually be beneficial, at least in the short term. It streamlines the genome, removing excess DNA. Or you could delete something valuable, which will decrease the net functionality of the genome. The costliness of this depends on how valuable that region is. Eyes in a cavefish don't serve much of a function anymore, so while a deletion in a developmental gene responsible for eye formation is technically breaking a functional region, it's not costly because eyes are no longer utilized by the fish. Mutations are often characterized by their rates. How often do deletions or duplications occur in a given genome per some unit time? Since both occur, there can actually be an equilibrium reached between these two values. If duplications occur in a region at a constant rate u and deletions at rate v, then the equilibrium expected frequency of duplicates maintained is a function of v over u plus v. If the rate of deletions exceeds that of duplications by, let's say, an order of magnitude, then there will be, on average, less than one duplicate maintained per individual. If duplications exceed that of deletions by a similar amount, then there will be approximately one duplicate maintained per individual. If these mutation rates exceeds the selective cost or the rate of genetic drift, then the duplicates persist just by mutation pressure alone. In fact, even if the presence of these duplicates are lethal, their expected frequency in the population will exactly equal the mutation rate, a finding first worked out algebraically by J.B.S. Haldane. The interaction between these different forces with respect to a specific organism is known as that organism's population genetic environment. environment. Contextualized this way, it helps us predict how evolutionary dynamics are expected to play out, given that we understand something about how big the population is and what kind of mutational pressures that population has. For example, if a population is relatively small and has a strong insertion bias, then we might expect that they would have large genomes, as selection is likely too inefficient to purge all the duplications being constantly supplied by mutation. Now, as stated, since there is a deletion pressure as well, an equilibrium will eventually be reached. The genome's not just going to expand forever, and if it did, the cost of the cell would eventually become so large that it just couldn't persist. At least comparatively, we'd expect this population to have a larger genome than one that has many individuals and a deletion bias. Okay, so we've been speaking sort of abstractly thus far, but now let's look at some actual biological data and see if we can justify the intuitions that we've built in this section. Before making broad predictions, the first thing we'd like to know is how costly excess DNA is to organisms. If any excess DNA has fitness effects that exceed that of drift, then we should never see any junk DNA. Selection should always purge it. However, if it turns out that possessing excess DNA is like a parasite that costs us a little, but not really enough to notice, then junk DNA is mathematically guaranteed given duplication and deletion rates alone. The first thing to note is that we understand in broad strokes the sorts of mutational biases that are present across the tree of life. Prokaryotes, archaea and bacteria, it turns out actually have a strong deletion bias. That is, genomic regions are more likely to get deleted than duplicated. Alternatively, most eukaryotes have been found to have an insertion bias. On average, DNA is duplicated in eukaryotic genomes far more often than it is deleted, and in fact, gene duplications have been found to happen at a very similar rate to just single nucleotide substitutions. Now, what's the cost of these, and can we make sense of why this dynamic may have evolved? Let's briefly talk through an important study by Michael Lynch and Georgy Marinov, published in 2015, that sought to estimate how costly gene insertions are to the cell. They estimated this by evaluating the number of ATPs used in replication, transcription, and translation for every known annotated gene relative to the total supply of ATP to that cell for a bacterial species, a unicellular eukaryote, a yeast, a nematode worm, C. elegans, and a weed, Arabidopsis. Importantly, they compared these costs to the population sizes of each of these species. When the cost exceeds that of the inverse of the population size, that is genetic drift, then selection is expected to dominate and should tightly regulate gene expression, including whether a redundant copy of a gene is expected to be tolerable. When the cost is less than or equal to that of the inverse of the population size, then selection is weak because there is an excess of ATPs relative to the amount needed to replicate, transcribe, or translate the gene. What they found was stunning. For bacteria, which we know have very small, streamlined genomes that are dense with protein-coding genes and little else, the cost of replication, translation, and transcription relative to their total cellular budget was very high, well exceeding the inverse of their population size, indicating that selection is tightly regulating these genes. It also means that any gene duplication would be very costly if it didn't immediately have a necessary function. Since this is incredibly rare, selection should be acting, on average, to purge duplicates in prokaryotes. These facts amazingly explain why prokaryotes have highly functional genomes with very, very little junk and genomes that are very, very small. Now, as we move from bacteria to eukaryotes, and especially multicellular eukaryotes like nematodes and weeds, the pattern reverses. In Arabidopsis, only translation is especially costly, whereas replication and transcription is just a small fraction of the total cellular budget. And this cost falls well below the inverse of the population size, again, falls well below the rate of drift, indicating that selection is expected to be incapable of tightly regulating it. This means that we should expect a few things. One, that duplicate genes can persist freely if they're only replicated or transcribed, but they don't get translated, as those two processes are very cheap relative to translation. And two, that transcription of existing but non-functional genes should actually be widespread. Again, because of how cheap the biochemical process is relative to the total cellular budget. And in fact, it is pretty costly to the cell to tightly regulate all those genes in the first place. And that cost, given its relative cost to the total cellular budget, is likely very low. These facts also help to explain how selection and drift have interacted to shape the mutation rate itself. As I said before, in eukaryotes there is an insertion bias, and this is permitted because excess DNA is not energetically costly. Furthermore, this excess DNA serves as a template for future tinkering and novelty, which may generate new functions. This helps explain why eukaryotes have gotten so complex, while bacteria, despite existing for billions of years, much longer than eukaryotes, have remained extremely simple and streamlined. Selection has tightly regulated their mutation rate to be both very low, much lower than eukaryotes, as well as being biased towards deletion because any insertion is immediately purged due to how costly it is to the cell. Hence, bacteria are pigeonholed into simplicity by selection itself. This may come as a surprise to many that complex multicellular animals and plants were only able to evolve because their population sizes shrunk enough to permit these duplicate genes to persist by drift alone, sort of flying under selection's radar, and hence stick around long enough for subsequent mutations to generate novel function. Indeed, increasing body size is believed to have played a major role in complexity by significantly reducing population size as organisms get bigger and bigger, and thus granting drift and mutation the opportunity to generate a ton of garbage that will eventually be co-opted into new functions. This garbage, or junk to put us back on topic, is absolutely necessary for complexity to evolve. New functions don't emerge out of nowhere. They typically have to be tinkered with from originally non-functional sequences. But that means these sequences must be allowed to persist, and if selection's too strong, as I just said, it will purge them. This leads us to a final prediction given what we've learned about the population genetic environment. If our intuition so far is true, then we should predict that there would be a linear relationship between genome size and population size, that extra genomic junk via duplications should be allowed to persist more and more as the population size gets smaller and smaller. Again, this is because drift and mutation are allowed to play proportionately larger roles when the populations are small. And lastly, that these genomes should not be getting larger with protein-coding genes or regulatory genes, but they should be expanding thanks to junk, or parasites, that is, transposons, pseudogenes, repetitive elements, etc. To cite Lynch once more, he and John Connery in 2003, and later in his 2007 book titled Origins of Genome Architecture, evaluated this relationship with a gigantic data set spanning across all domains of life. What they found was striking: a clear negative relationship between population size and genome size, and that as genomes got larger, the fraction of it devoted to proteins and regulatory regions shrank rapidly. To reiterate, this pattern is only expected given the allowed proliferation of non-functional genomic elements, aka junk. This junk expands via mutation, is allowed to persist thanks to drift overwhelming selection, but ultimately serves as the template for subsequent tinkering and novelty. It does so in redundant, wasteful ways, but this is what is expected of systems that are not being fine-tuned by selection or some intelligent agent, but rather being allowed to bloat by mutation and drift. These observations, coupled with an understanding of the population genetic environment, beautifully explains the so-called C-value paradox. It turns out complexity is not expected to predict genome size. Instead, genome size is a function of the rate of genetic drift and the mutation rate. And again, this relationship can only come about if the bulk of the genome is composed of junk DNA. We've now discussed how genome size varies across the tree of life, how this is explained by population genetics, and excellently predicts the observed data, and how the genome is not only a blueprint for building an organism, but it's an environment itself that is capable of being invaded by different kinds of parasites. Hopefully these facts help you understand why junk DNA is virtually guaranteed to exist, both theoretically and mathematically, given like the rates of mutations that we've observed, the sorts of biases and mutation rates we've observed, and the bioenergetic cost of gene insertions relative to the rates of genetic drift. But we're really only just beginning. There's a long history of the concept of junk DNA that has been utterly contorted, misconstrued, and misrepresented for the past 2 decades. News articles, interviews, and even scientific publications have all proclaimed junk DNA either a dead idea or one that was built on our own ignorance. Our discussion thus far should show that junk DNA is not built on ignorance, but on a combination of comparative genomics, biochemistry, and population genetics, and that as a prediction, it fits perfectly the data at hand. But let's spend some time on how junk DNA as a concept first emerged, and then we'll discuss why so many people, again, including scientists themselves, seem to be so confused by it. The concept of junk DNA predates the discovery of DNA itself. It really began with the work by population geneticist J.B.S. Haldane and subsequent work on X-rays and the human mutation rate by Nobel laureate Hermann Mueller. In 1937, Haldane published a paper that evaluated the theoretical fitness consequences of mutations, noting that even lethal mutation should persist in populations due to recurrent mutation. This is due to the fact that even in infinite populations, an equilibrium will eventually be reached between natural selection's ability to purge a mutation and the rate at which that mutation keeps getting added back to the population. Due to this, all populations are expected to harbor some amount of deleterious, even lethal, genetic variation. In fact, in diploids, this variation is expected to be higher as it can be effectively masked by heterozygotes. This concept became known as the genetic load, the reduction in fitness of a population due to recurrent mutation. In the decades since Haldane's calculations, many additional types of mutational loads have been identified, including what is called the substitutional load, which is the cost in terms of genetic deaths in a population where natural selection is trying to spread one allele at the cost of another. Another is the drift load, which is the persistence of mildly deleterious alleles in small populations due to the inefficiency of selection to purge them. Ultimately, Haldane's contribution to the eventual concept of junk DNA was pointing out that recurrent mutations decrease the fitness of the population, and that there is some theoretical number of mutations that can be tolerated before you reduce fitness so much that the population cannot replace itself. It just cannot persist. It's important to note here that Haldane's model and all subsequent models of the genetic load rely on a few strong assumptions about the nature of selection. That is, they all assume that selection is hard. This means that it's impacting the population growth rate by impacting survival itself. So if a population accumulates too many mutations that impact organisms' ability to survive, it enters this sort of extinction vortex because it's no longer capable of replacing the previous generation via births in the subsequent generation. But mutational effects can also be soft. This means that their fitness effects is in respect to other members of the population, often mediated by competition. The best way to think about this sort of latter form of selection is a little crude, but I think it's apt. So imagine a bunch of individuals being chased by a tiger. The tiger can only eat one of you. It only has so much room in its belly, right? So all you have to do is be faster than the slowest person. It isn't necessary that you be the fastest person in the population, and hence selection weeds out individuals based on fitness effects relative to others, not in an absolute sense. And indeed, if mutations happen such that all of you were now incredibly slow and none of you could outrun one another, the tiger is still only going to eat one of you. This point is quite important when we start thinking about how to resolve the so-called mutation load paradox, but that's a topic for another video that I've covered previously, links in the description below for that. So Haldane showed a genetic load was theoretically possible, and even could be used to estimate the human mutation rate. Side note, his estimate was remarkably close to the rate that we know today, despite not knowing anything about molecular biology of mutations, or even that DNA was the heritable material. Next comes Hermann Muller, who studied how X-rays cause mutations in laboratory populations. In 1950, he gave an incredibly influential speech titled Our Load of Mutations, in which he suggested the mutational load in humans was remarkably high. He argued in subsequent papers that given this mutational load, the number of mutational targets in the human genome capable of reducing fitness must be no more than 30,000. A way to think about this number is that there could really only be about 30,000 genes, protein coding or otherwise. Again, they didn't have any conception of the molecular biology of genes at this point. That could be functional in the sense that they influence survival and reproduction. Any region that does not influence survival and reproduction can mutate freely, and so is irrelevant to the genetic load. Subsequent biologists came to the same conclusion, including famed molecular biologists like Thomas Jukes and Sidney Brenner, the latter of which elucidated the genetic code and won a Nobel Prize for his work in C. elegans. So the prediction by the 1960s by evolutionary biologists was that there would be no more than around 30,000 genes in the human genome, given the prohibitively high human mutation rate. The remarkable accuracy of this prediction, given what we now know about the human genome, is criminally underappreciated. That these biologists in the 60s predicted this number without the need for billions of dollars in NIH money and fancy modern molecular labs is a remarkable fact that every biology student should be taught. Thus far, we've mostly focused on theoretical results, or at least results about empirical data that are interpreted through the lens of existing theory. The first empirical evidence of junk DNA came from a technique called COD analysis. COD analysis belongs to a class of molecular techniques called reassociation kinetics. It involves heating the DNA such that it denatures it into single strands, and then measuring the amount of time it takes for the two strands to completely reassociate. What you learn from this is how much of the DNA is repetitive. Regions that exist in single copies take a long time to find their complement, and so reassociate very slowly, whereas those that have many copies associate very quickly. This produces what is called a COT curve, and these curves can be compared across species. For E. coli, it was found that most of their genome existed as single copy, unique sequences, and so took longer to reassociate than the genomes of vertebrates like mice, because so much of their genome was repetitive and so reassociated very rapidly. For eukaryotic genomes, they found that large portions of it was repetitive. This helped explain the curious observation mentioned at the beginning of this video that some organisms had lots of DNA in their cells, while others, seemingly more complicated, had much less. If most of this repetitive DNA was non-functional, then it beautifully explained the disparity between genome size and complexity. It should be noted that the lab that popularized the COT technique was led by Roy John Britton, who was a bitter opponent of the junk DNA hypothesis. In 1968, he wrote, quote, "A concept that is repugnant to us is that about half of the DNA of higher organisms is trivial or permanently inert on an evolutionary timescale." A similar technique that was popularized around this time was DNA-RNA hybridization, which sort of follows a similar logic. This is a molecular technique that involves taking RNA transcripts from a cell and then hybridizing them back onto the DNA after it had been denatured. The purpose of this technique was to identify how much of the DNA was actively involved in producing messenger RNA transcripts and hence could presumably be translated. So this was a way of measuring how many protein coding genes could be in the genome. What researchers found was that despite the large number of transcripts produced, most of them hybridized to very tiny proportions of the genome. That is, very little of the genome was dedicated to producing proteins, which corroborated the COD analysis that perhaps only a small fraction of the genome was functional. It should be noted that while some took DNA-RNA hybridization as evidence for widespread junk, there were many early critics. In 1973, S.J. O'Brien argued that while these analyses suggested that most of the genome wasn't actively involved in directly generating proteins, it couldn't tell us how much was involved in regulation or was structurally important, and that we should be cautious about assuming the rest of it was junk, or that we can assign an exact number to just how much junk there is. Thus, two observations early on supported junk DNA. First, theory on the mutation load and observations of the mutation rate suggested that most organisms could not have a very high proportion of functional DNA, or they would rapidly go extinct due to mutation pressure. And secondly, that COD analysis seemed to suggest that most of the genome was repetitive in multicellular eukaryotes, which helped explain why there seemed to be a lack of correspondence between complexity and total cellular DNA. But where did the term first come from, and when did it attain widespread acceptance? Its origin is unclear, but some have suggested that it was being used colloquially as early as the 1950s. But it was formalized and popularized by Susumu Ohno, who gave an influential speech in 1972 titled "So Much Junk in the Genome" to the Brookhaven Institute. Ohno's argument is rarely given breath, despite just how brilliant it was, and it's worth hashing out here. Ohno is responsible for, and rarely credited for, our modern conception of how new genes emerge. He wrote a highly influential book titled Evolution by Gene Duplication, which argued that new functions in the genome first emerge by duplication and then subsequent tinkering, as I was talking about earlier. Importantly, he pointed out that most of the time, duplicate genes are evolutionary dead ends. They rapidly degrade by mutation, becoming non-functional pseudogenes. Occasionally, some of them will acquire a novel function and be favored by natural selection, but this is rare relative to the frequency of gene duplication. As I said earlier, in multicellular eukaryotes, we know that gene duplication happens almost as often as single base pair mutations. So again, I cannot stress enough that he wrote this long before we understood that there is an insertion bias in eukaryotes, or really had much idea about the general genomic architecture in animals. And yet he had incredible foresight. He knew that most mutations would degrade function, not create it, and because of this, most duplications and subsequent tinkering would produce nonsense. From this, he argued that most of the genome was probably junk, by which he meant non-functional. He did not mean harmful, or even necessarily useless forever. He meant merely that it didn't do anything of a functional relevance for the survival and reproduction of the organism. Sidney Brenner has likened this to having a bunch of random stuff in your garage. It doesn't really harm you outside of taking up some space in your garage, and indeed, every once in a while you might rummage through it and find some use for some of it. But mostly it just sits there. Occasionally you'll throw some of it out, but it's not doing anything for you one way or the other. As we discussed in the previous section, what is junk in one species will be actively purged in another, so it's important to never forget the population genetic environment when we're discussing what is and is not junk. Greyer has suggested distinguishing between these by using junk as elements that are useless but not harmful, by which he means not seen by selection, and garbage, which are actively harmful and expected to eventually be purged by selection. It's also important to note that Ohno was not referring to all non-coding DNA. He discussed at length transfer RNAs, ribosomal RNAs, other sorts of regulatory elements, was familiar with centromeres, etc. At no point were the proponents of junk DNA confused that if it didn't code for proteins, it must be junk. This is one of the many false narratives that have been promoted by many anti-junk ideologues. Junk DNA has always referred to non-functional elements, not non-coding DNA writ large. Most biologists, even critics of junk DNA, would agree that new genes emerge via duplication. But to contend that the origin of new genes is duplication, but there is no or little junk DNA, implies that natural selection will always find a use for redundant copies. This is at its core an adaptationist claim, that natural selection is some sort of all-powerful agent and will always win out over genetic drift and mutation, which acts to degrade duplicates. It also implies that mutation rarely breaks redundant copies, or that redundant copies somehow immediately have a use, and so selection weeds out mutations that break them. These ideas are contradictory to me. Biologists cannot simultaneously claim that the origin of new genes is mostly via duplication and there is no junk. That is fundamentally not how we understand these processes to work, either at the empirical, observational level, or theoretically. Another strange rewriting of history is the claim that all biologists were on board with junk DNA, and that the concept itself actively set back genomic research. I've already given examples of biologists that were pretty anti this idea, but there's a telling letter that Thomas Jukes wrote to Francis Crick after Francis Crick suggested that most of the genome was infected by selfish DNA. Jukes wrote, quote, "Dear Francis, I am sure that you realize how frightfully angry a lot of people will be if you say that much of the DNA is junk. The geneticists will be angry because they think DNA is sacred. The Darwinian evolutionists will be outraged because they believe every change in DNA that is accepted in evolution is necessarily an adaptive change. To suggest anything else is an insult to the sacred memory of Darwin." Also in the 1970s, we discovered that eukaryotic genes appeared to be in pieces. There were small regions of them that served as templates for the amino acid chain, whereas much of the stuff between was snipped out and degraded. Walter Gilbert, in a 1978 article titled "Why Genes in Pieces," proposed calling the regions that are translated exons and those that are snipped out introns. While many, including Ohno, considered these introns to be junk, this view was not universal. Gilbert actually argued that genes evolved to be in pieces to permit them to be assembled in different ways, and hence a protein-coding gene could code for many different kinds of proteins depending on how the transcript was assembled. What he had hit upon was later confirmed to exist and called alternative splicing. It's important to note that the existence of alternative splicing does not mean that introns are not junk. If the purpose of alternative splicing is to generate novel combinations, then you need a maximum of a pair of splice sites, but introns can be thousands of base pairs long. Indeed, the coding parts of genes are tiny islands in a sea of repetitive DNA destined for degradation. Since introns are merely spliced out, there's no pressure for them not to expand in size. This expansion doesn't increase the function of alternative splicing. It's merely the passive proliferation of junk, given that we know eukaryotes possess an mutational insertion bias. Why did Gilbert and others feel the need to come up with functions for introns and other repetitive DNA that seem so obviously junk? The historical context to remember here is that prior to the 1970s, adaptationism reigned supreme in biology. Adaptationism, as I mentioned a moment ago, is the idea that all features of organisms have been shaped and perfected by natural selection, that any mutation that did not have a positive effect on fitness would be purged. Leading evolutionary biologists came to accept this position, such as Ernst Mayr, who argued in his 1963 book, Animal Species and Evolution, that no genetic element that was initially neutral would remain so for long, that selection would find a use or would discard it. Thomas Cavalier Smith, who is responsible for much of the taxonomy of protists, also opposed the idea of junk DNA, arguing instead that differences in genome size played a cytoskeletal role in the cell and was itself shaped by natural selection. These ideas faced a crisis in the late 1960s when it became clear that organisms harbored a startling amount of genetic diversity. How could this be if selection was constantly sifting through mutations, picking out the best ones? Shouldn't organisms be generally genetically uniform? In 1968, population geneticist Moto Kimura provided the answer to this conundrum when he proposed what would become known as the neutral theory of molecular evolution, that most of this genetic variation was neutral and did not impact fitness at all. In fact, he went so far as to argue that genetic drift was the dominant form of evolution at the molecular level, that most mutations have little or no impact on fitness, and the rate at which they were lost or fixed in a population was a function of that population's size. This concept was supported from a biochemical perspective in 1969 by Jack King and Thomas Jukes, who argued that amino acid changes appeared to have no impact on the function of several genes, mostly focused on the mitochondria. This led to the infamous neutralist selectionist debates of the 70s and 80s, in which evolutionary biologists battled it out over the relative roles of selection and drift. This debate still goes on to a lesser extent to this day, but the major result to come out of this period was Tomoka Oda's, a postdoc of Kimura's, theory that most mutations were slightly harmful, not strictly neutral, and that they could persist so long as their harm was so small that selection didn't take notice. This was an important update from Kimura's work because it didn't make any sense to many that genomic expansions via parasitic DNA could be strictly neutral. It must be causing some harm. And if it was causing harm, how could it still be there? Oda provided the answer in her nearly neutral theory of molecular evolution. They persist for the same reason you can harbor many other kinds of parasites. They harm you, but not really enough to stop you from living your life. Another important discovery that was initially unappreciated was made by Barbara McClintock in the 1950s. As I said before, researchers recognized that the majority of the genome didn't appear to hybridize with messenger RNA and was highly repetitive. But what was it? How did it get there? Could all this be structurally important or somehow involved in regulation, as O'Brien thought? McClintock discovered that a large part of the genome was what was initially called jumping genes. Genes that could move around in the genome, causing all sorts of wacky phenotypic effects, most of which were quite harmful. These are, of course, transposons, which we have talked about at length. They are the parasitic DNA elements that make up most of the typical eukaryotic genome. Francis Crick would go on to characterize them as selfish DNA, an idea that would later be popularized by Richard Dawkins in his 1976 book, The Selfish Gene. We now know transposons and endogenous retroviral sequences make up at least 60% of your genome. Given the strong evidence that introns are actually derived from transposons, pushes that number to greater than 80%. The 1980s was a revolutionary time for developmental biology, especially in the discovery of a handful of genes that appeared to be responsible for regulating development. These genes, called Hox genes, were found to exist across the tree of life, and subtle changes to them appeared to explain the difference in body plans between organisms as distant as fruit flies and humans. That so few genes were needed to create radically different body plans meant that you don't need a ton of DNA differences to explain why fruit flies and humans are different. You just need a handful of changes in how development is regulated, when certain genes are expressed, and how much of a gene product is produced, and all of this can be achieved with minor alterations to the DNA. This lent additional support to the notion of widespread junk DNA, as it provided a simple mechanism for explaining complex organismal body plans that didn't rely on adding more DNA. Thus, by the 1990s, most evolutionary biologists were convinced of the existence of junk DNA. There were good theoretical reasons to believe that it existed, the genetic load and neutral theory, and plenty of observational evidence in the form of transposons, introns, vast differences in genome size between even closely related organisms, and the general lack of correspondence between gene number and genome size. But then something bizarre happened around the time of the Human Genome Project that has continued ever since. The popular narrative began to shift. Suddenly, the concept of junk DNA came under attack, and in the strangest way. The historical narrative shifted from "we have good reasons to think junk exists" to "it was based on ignorance." Scientists and journalists unfamiliar with population genetics and evolutionary theory proclaimed that we always thought junk DNA was all the non-coding DNA, despite the fact that Ohno, who popularized the term, wrote extensively about non-coding genes. All of this culminated in 2012 with the publication of the ENCODE results, with one news article titled, quote, "ENCODE Project Writes Eulogy for Junk DNA." The ENCODE project may be the biggest scientific controversy in my lifetime. Definitely in my specific field of evolutionary biology, and since it's the centerpiece of the modern debate of junk DNA, we should spend some time talking about it. What it is, how the project came to be, what it found and presented, and why it caused such a firestorm in the community. In 1915, R.A. Fisher, who would become one of the founders of theoretical population genetics, in a critique of pre-adaptationism proposed by a systematist named Ohno, wrote, quote, "The tendency of modern scientific teaching is to neglect the great books, to lay far too much stress upon relatively unimportant modern work, and to present masses of detail of doubtful truth and questionable weight in such a way as to obscure principles." In the age of the internet and clickbait culture, this critique is even more poignant. We have become obsessed with flashy new things, especially if they are framed as challenging the way we thought the world worked. How many times have you seen a news article that contains something in the headline like, "New discovery overturns old theory"? We love banishing old ideas, trashing people of the past, and in general, assuming that we are the enlightened ones because we have access to the newest technology. But in failing to read the classic work that came before us, we often erect straw men of past ideas, which make it easy for us to say, look, modern man's smarter than man in the 1950s. Biochemist Larry Moran, in his recent book, What's in Your Genome, promotes this phrase paradigm shaft to characterize this. A paradigm shaft is a play on a paradigm shift, but instead of shifting the true paradigm, a paradigm shaft is when you make up a paradigm that no one believes in and then claim to overturn it. This has been what has happened with junk DNA. Critics claim that junk DNA was based on ignorance, we just didn't know what these things did and so assumed they were junk, and that proponents of junk DNA assumed all non-coding DNA was junk. By providing first the history of junk DNA, I hope you are well equipped now to see how much of a paradigm shaft this is. Okay, so what was the ENCODE project? It stands for the Encyclopedia of DNA Elements and has the goal of uncovering biochemical activity in the human genome. It came off of the back of the Human Genome Project, which sought simply to sequence the entire human genome. Both are projects of the National Institute of Health, a massive federally funded agency in the United States dedicated to research into human health. My position is actually funded by the NIH, as are the research labs of thousands of biologists. ENCODE started as 8 research groups during its pilot phase, but after 2007 has grown to over 400 scientists working across 32 research labs around the world. The entire project has nearly $300 million dedicated to it, a massive sum of money for scientific research. The main goal of ENCODE was to clarify a puzzling fact of the Human Genome Project, that only around 2% of the genome appears to code for proteins. A few more percent were known regulatory elements, tRNA and ribosomal RNA genes, etc., but that pushed the known functional fraction to only around 5% going into 2005. While this was no surprise to evolutionary biologists, as we discussed in the historical section, to cellular and molecular biologists who often seem rather poorly trained in evolutionary biology, this was quite a shock. Surely all that DNA must be necessary and special, hidden somewhere in the genomic dark matter, as some silly journalist took to calling it, must be the secret to being human. I want to pause for a moment and give a shout out to an excellent little collection of essays by evolutionary biologist Richard Lewontin called Biology as Ideology. In this, he critiques the promise of the Human Genome Project as a reductionistic view of biology, that somehow everything about being human, including understanding all facets of human disease, can be boiled down to nucleotides. The promise of both the Human Genome Project and ENCODE was to improve human health, but neither of those promises have been met, despite the millions of dollars poured into them. Why? Because most human diseases aren't the results of genetics, for one. For two, it turns out that most phenotypes are complex combinations of genetic and environmental effects, and that genomic context may play as big a role as specific nucleotides themselves. These projects also ignore developmental stochasticity, cultural inheritance, indirect genetic effects, etc. My point is that no matter how hard you search, there will simply be a great number of phenotypes that will never be straightforwardly assigned to some part of the genome. It's a gross oversimplification of biology to think there would be. What makes us human expands far beyond just our genes. But I digress. Cellular molecular biologists love reducing things down to their component parts, and the promise of ENCODE was that it would uncover what the other 95% of our genome was doing. And they began with an a priori assumption that it must be doing something. The project began with a pilot phase, which worked with only 1% of the genome to test their methods and ability to identify biochemical activity. Their main focus was on transcription factor binding sites, histone modification, chromatin structure, and transcription levels. This pilot phase, the results of which were published in 2007, found a ton of novel transcripts that weren't associated with protein-coding genes. They also found transcription start sites that were not associated with any known protein, but that interacted with chromatin structure. For a brief refresher, the genome is a three-dimensional molecule that is variously wound around a series of proteins called histones, and regions that are tightly wound are referred to as heterochromatin. These regions cannot be transcribed because they prevent transcription factors from binding to the DNA. It's too tightly wound and thus inaccessible. Accessible DNA for transcription exists in euchromatin regions, which are not tightly wound. These two classifications are not consistent across time and cell stages. Sometimes regions are in heterochromatin and sometimes euchromatin, depending on when the cell needs certain things. This condition is called facultative heterochromatin to distinguish it from constitutive heterochromatin, the latter of which is virtually always in the heterochromatic state. This includes highly repetitive regions, especially those on human chromosomes 1, 9, and 16, as well as the Y chromosome, and is densely concentrated around telomeres and centromeres. This latter fact indicates that heterochromatin, in addition to keeping certain regions silenced, also plays a role in chromosome stability. So transcription factors that interact with chromatin may cause it to relax or contract, permitting transcription to occur. As expected, the pilot study found histone modification and chromatin accessibility strongly predicted the presence of a transcription start site. Lastly, they found, consistent with previous studies, that only around 5% of the human genome shows signs of evolutionary constraint, and that of these, around 60% showed identifiable biochemical function. Evolutionary constraint, as discussed earlier, means simply that across species, this region is highly consistent in its structure and sequence. This means that many species are utilizing this region in exactly the same way, which is expected if it is functionally relevant to the organism's health. Of course, the ENCODE project suggested that much more of the genome was likely transcribed, interacted with histones and chromatin structure, etc., than what was predicted based on evolutionary conservation alone. While this ruffled some feathers in 2007, given that they only worked with 1% of the genome, they did not yet have the power to really challenge the junk DNA hypothesis. That changed in 2012 with the publication of an onslaught of papers that resulted from the second phase of the project. In total, there were 30 papers, 6 in Nature, 6 in genome biology, and 18 in genome research. This phase utilized 147 different cell lines from many different tissue types, reporting the results of biochemical activity, as in the pilot phase, that occurred in at least one of these cell types. The results were hailed as ringing the death knell for junk DNA. They found that 80.4% of the human genome participated in at least one biochemical interaction, which they defined as being transcribed or associated with a histone, that is, was capable of modifying chromatin structure. They found that greater than 95% of the genome was nearby to at least one biochemically active region, as identified by ENCODE, which they take as evidence that most of the genome could be interacting in some functional way. They found nearly 400,000 regions that showed enhancer-like functions. Enhancers are regions of the genome that bind proteins that increase the likelihood of transcription. Another 70,000 acted as promoters, which actively recruit transcription factors. Lastly, they used a genome-wide association map. Studies of this type are called GWAS, to identify variable regions associated with disease phenotypes. They found found that most diseases were enriched in non-coding DNA, especially associated with transcription factors. The flurry of technical publications, themselves quite restrained, was matched by an even greater number of news reports, interviews with leading researchers, and promotional materials. Yes, they literally produced promotional videos and articles like they were trying to sell a product.
A decade ago, scientists broke down the 3 billion letters in an average human genome. Now they're trying to translate those letters into a comprehensive instruction manual for building a person.
But also all transcripts, so all of the RNA that comes off of the genome. And very importantly, the project aims to characterize the regulatory parts of the genome. So a striking overall result that the ENCODE project reports is that they can assign a function, a biochemical function, to 80% of the human genome. The reason why this is striking is because not such a long time ago, we still considered that vast proportion of the human genome was simply junk. Because of course we know that it's only 3% that encodes proteins.
The level of hype was incredible, but the message that rang louder than all others was ENCODE has discovered biochemical activity for 80% of the genome, and it turns out that the idea of junk DNA was wrong after all. I hope that by discussing what junk DNA is, framing it in light of parasitism, and discussing the history of the concept prior to telling you ENCODE's results, has situated you to immediately recognize the problem that these PhD biochemists and molecular biologists apparently missed. For them, ENCODE was a vindication of what they always thought. DNA is there, it must be doing something. Oh look, turns out that biomolecule does biochemistry. Since biomolecule does biochemistry, biomolecule is functional, and its function is to do biochemistry. Ta-da! See the problem? Parasites are rarely passive passengers. Biomolecules will absolutely do things if they're in an environment in which they can interact with other molecules. The feverish excitement over biochemical activity is akin to becoming excited to discover that your shoe, despite long degrading to no longer protect your feet, still manages to bind gum. The response to ENCODE, and frankly the irresponsible journalistic excitement over it, was swift and vicious, and rightfully so. Here are a few papers that immediately attacked ENCODE on a combination of theoretical, empirical, statistical, and technical basis. Let's chat through each of these, some of which will be rehashes of what we've already stated, but when it comes to learning, it's all about repetition. First, let's talk some technical issues. It's often forgotten that the cell lines used by ENCODE were mostly pluripotent stem cells and cancer cells, two cell types that are well known for being transcriptionally permissive. Past work has shown that these cells can have transcription levels 1,000 times higher than other cells, with transcription occurring from even very weak promoter sequences. Very weak promoter sequences means any sequence that even slightly resembles a true promoter sequence. In addition, they use HeLa cells, which were originally derived from human cancer cells but have grown exclusively in laboratories since the 1950s. They have diverged so extensively from normal human cells that some have even proposed calling them a new species entirely. Second, let's chat through some empirical expectations. There are several junk DNA elements that are expected to be transcribed but not functional. Transposons are excellent examples of this. They have their own promoters, transcription start and stop sites, and even several genes that they code for. All of this is necessary for them to function as genomic parasites. Given that they occupy greater than 50% of the human genome, it's not at all surprising to find high levels of transcription. Even transposons that have been mutated, such that they are no longer capable of transpositioning, may still harbor promoter sequences that will occasionally recruit transcription factors. These will result in short RNA transcripts that have no function and will simply be degraded by the cell. Another is introns. We know introns are transcribed, and these comprise nearly 30% of the human genome. While some are known to be spliced out and form functional RNAs, the vast majority are degraded after splicing. This is especially obvious when you consider how variable intronic sequences are at the sequence level between even two humans. Between you and I, there can be immense differences in the sizes of our introns and what sequences they possess. This is because while the presence of introns are necessary, maybe, that is to some extent debatable, their sequence and size doesn't matter. You can technically get away with just splice sites. Everything in between is quite literally junk. You could toss it out and still function fine. How do we know? Because between you and I, the total amount of intronic DNA will be different. Nature has literally performed the experiment for us. It's unethical to perform lab knockout experiments on humans, but when this was performed in yeast, only 3% of knocked out introns and across 87 genes yielded any phenotypic effect. This is consistent with most introns being junk. Lastly, pseudogenes are expected to be transcribed and even occasionally translated. I'll give a specific example. Shovel bugs, the little crustaceans I talked about at the top of this video, are blind. They have no pigment in their eyes, but they still have eyes. Why? Why bother putting resources into forming them at all? The genes that code for eyes are pseudogenes. They don't function correctly because they don't form functional eyes, but they still build these vestigial structures. Hence, they are transcribed and translated, but there is no reason for them to be outside of historical contingency. A pseudogene is one that once had a function, but due to mutation, that function has been lost. We do not expect a pseudogene to just immediately stop being transcribed. If a promoter is intact, the cell doesn't know that what it's transcribing isn't going to function. Thus, the amount of transcribed DNA, given the estimated proportion of transposons, introns, and pseudogenes prior to ENCODE, is not at all surprising, nor does it have any bearing on whether junk DNA exists. Transcription is not de facto evidence of function. On the empirical front, we have many studies documenting spurious transcription. In particular, several studies show in yeast that even synthetic, random DNA inserted into a yeast genome will become transcribed. Furthermore, it has long been known that many sequences can interact with chromatin structure and bind to histones without being functionally significant. One study in 2010 found that out of 8,436 possible histone modifications, only 142 of them, or less than 2%, had any functional relevance. What kind of functions did ENCODE assign to histone modifications? One was, quote, preference for the 5' end of genes. Grauer and colleagues write, this is akin to asserting that the function of the White House is to occupy the lot of land at the 1600 block of Pennsylvania Avenue in Washington, D.C. Another ENCODE function prescribed for a great deal of the genome was methylation. To ENCODE, if a region is methylated, it must be functional because methylation is a kind of gene regulation. Specifically, it can silence genes. But this is extremely odd because methylation is merely a property of chemistry. In the genome, what are called CpG sites are strings of cytosines and guanines next to one another. Whenever you have such strings, they possess the capacity to be methylated. In particular, cytosine readily methylates to 5-methylcytosine. This propensity has also caused what are known as CpG islands due to the fact that deamination of 5-methylcytosine results in a thymine, leading to a TG mismatch, which is often repaired to an AT instead of the original CG. Over time, this causes a depletion of CpG sites. Each of these are simply chemical reactions. None of this has necessarily any functional significance. Again, the ability to be methylated is not itself a function. It's merely a side effect of having a given chemical structure. Now let's talk statistical issues. ENCODE consistently favored sensitivity over specificity. What this means is that they relax their statistical evaluations to permit identifying the largest number of possible elements. That is, they made their stats overly sensitive, which increases the chance of false positives, but ensures that you don't miss anything. A specific example is when they elevated their p-values to 10% instead of the standard 5% in every other scientific study. P-values are a measure of the accepted rate of false positives. Typically, you want this to be less than 5%, but ENCODE, for reasons unknown, accepted anything less than 10%, twice the standard statistical practice. Again, this means that they necessarily identified a greater number of associations than would be typically accepted, ensuring that their estimated functional fraction was higher than any other study to date. They even complain in one of the 2012 papers that a 10% threshold may be too stringent. Increasing it would allow identifying even more function. Apparently, ENCODE researchers are so dedicated to finding function that they're willing to suffer from a kind of genomic apophenia, accepting patterns that aren't real simply because you think some of them must be real. Again, Grauer and colleagues in their 2012 critique stated that if ENCODE's goal was simply to identify all possible functional elements, irrespective of how many of them were false positives, they could have saved $300 million by just proclaiming the entire genome was functional. Not a single element would have been missed. The last thing I'd like to discuss regarding ENCODE is the implicit assertion that function can be maintained in the complete absence of purifying selection. As I said before, mutations constantly erode function in one way or the other, and so to maintain function, selection must remove harmful variation. That only 5% of the genome shows signs of evolutionary constraint is strong evidence that it must mostly be junk. To insist otherwise is to claim two things. One, that 95% of the genome has acquired a unique function since we split from chimpanzees a mere 6 million years ago. This would require mutation rates orders of magnitude higher than observed, and that nearly all mutations are beneficial, constructing new functions far more rapidly than what has ever been observed in any lab or agricultural setting. And two, that these functions can exist entirely without the need for selection to protect them from mutation. That is, they are immune from degrading. Lineage-specific functional sequences are the toughest to identify selective constraint for because they can still have large amounts of polymorphisms if they are still in the process of being purified by selection. But given no mutation rates and distributions of fitness effects, which overwhelmingly show that most mutations are deleterious, to explain the remaining 95% of the genome that shows no selective constraint as being the result of lineage-specific novel gene acquisition is akin to invoking a miracle. In 2014, after a dozen or so critiques of ENCODE's 2012 results, a much more careful and thoughtful publication by some of the leading ENCODE researchers walked back much of their claims in the press 2 years prior. They argued that when it comes to biological function, a more nuanced definition is needed than what they used, and that a great many of the biochemically active regions of the genome may indeed have no functional relevance. By that time, several of them had set their expectations on total genome functionality at a mere 20%, clearly leaving the vast majority of the genome, 80% of it at least, as junk. So much for ENCODE writing the eulogy for junk DNA. As I was working on this script, a new paper came out documenting the largest genome ever recorded. This small, unassuming fern has a genome that is 160 billion bases long. That's more than 50 times the size of the human genome. The largest record was previously held by Paris japonica, a flowering plant with a genome of 149 billion bases, and dwarfs the marbled lungfish, the largest animal genome of 133 billion bases. Massive, multi-million-dollar projects like ENCODE are constructed under the assumption that the vast majority of the genome of any organism, not just humans, is functional. To claim that there's something special about the human genome size of 3 billion bases, that it exists in some sort of Goldilocks zone where it's entirely functional, but any bigger would be junk, is to ignore how many organisms with much smaller genomes get along just fine. After all, birds have just as many cell and tissue types as we do, as well as extremely complex behavior, and rarely have genomes larger than 2 billion bases. I hope that by framing junk DNA within the context of ecology, by thinking about it as a parasite and the genome as a kind of environment, served as a useful way of conceptualizing why junk DNA spreads. You should now also be equipped to answer challenges about how junk DNA should have been purged by selection. In fact, population genetics predicts widespread junk DNA, especially in multicellular eukaryotes, and this is corroborated by studies showing how cheap transcriptional noise really is. Further, we dispelled the many myths perpetuated by some biologists and creationists that junk DNA was never challenged historically, that it resulted from ignorance instead of data, and that it was a science stopper. It sure didn't stop the US government from dumping nearly $300 million into dozens of research labs around the world for the ENCODE project, or stop the $2.7 billion that it forked out for the Human Genome Project. And this massive amount of money came with a heavy burden. These labs needed to show results. Can you imagine if all that money had been spent and they found what we already knew, that most of the genome was junk? The incentive to find as much function as possible caused them to prioritize statistical sensitivity over specificity, which resulted in an inordinate amount of false positives. And lastly, we talked through the ENCODE project, its results, its many transgressions, and why the group was forced to roll back its initial sensationalist claims to settle on a more reasonable, though I still think over-generous, functional fraction of 20%. Junk DNA is not an idea born of ignorance, but an incredibly powerful hypothesis that makes sense of many observations in nature. It has strong theoretical backing as well as decades of empirical support. That it challenges the sensibilities of some molecular biologists and a whole lot of creationists is an unfortunate side effect, but that is ultimately irrelevant to its truth. Thanks so much for being here. Lots of links in the description below, as this video leaned heavily on at least a dozen papers. I've also included links to the ongoing back and forth between Dr. Dan of Creation Myths and the Discovery Institute, which is sort of what prompted me to revisit this topic in the first place. Again, if you have any questions, comments, thoughts, criticisms, drop them in the comments section below. Thanks again for being here, and I will catch you next time.